A scientist's journey to alter gene expression. Not too long ago, there was a scientist who wanted to alter the gene expression of the crown imperial flower. Whenever the scientist walked out into his garden, the first thing he always smelled was the terrible odor the flower gives off. He decided he wants to alter the gene expression for the pungent smell of the flower in order to remove the odor from his garden. The scientist knew that this process would involve transcription. He knew that transcription was the process of using an RNA polymerase II to create an mRNA strand from DNA that will be translated into a protein. The scientist did a lot of research on how to reduce or completely eliminate the expression of the odor gene in the crown imperial. However, he could not find any strains of crown imperial that had a mutated version of the odor gene. He finally came about two options to alter the expression of the gene: CRISPR or RNAi. How exactly do these methods work, the scientist asked. In CRISPR, we take a piece of guide RNA that matches the DNA sequence we want to mutate and mix it with Cas9, which will cut out the sequence. We inject this mixture into healthy crown imperial cells. The guide RNA Cas9 complex then locks onto the PAM sequence. Once the guide RNA recognizes the DNA sequence, we unzip our DNA and have Cas9 cut out our sequence that has the gene for the odor. The DNA tries to bind back together but is now disabled. We can add a healthy DNA with the sequence we want to repair the DNA. We now have our mutated DNA sequence that contains the genes we want. In RNAi, we can use either miRNAs or siRNAs to achieve our goal. For miRNA, we use DNA, undergo transcription to form pre-miRNA, cleave the hairpin structure, and form a single miRNA using a dicer, which combines with the protein to form the RIS complex. This imperfectly attaches to the mRNA and inhibits translation. For siRNA, we use double-stranded RNA with the dicer to form siRNAs. These siRNAs then attach to a protein and form the RIS complex, which attaches to mRNA and cleaves the mRNA, degrading it. Now that he knew how each method worked, it was time to decide which method was better by examining their similarities and differences. Now both are methods of altering gene expression. CRISPR could edit the gene, while RNAi could knock down the gene. It may not fully inhibit expression. CRISPR also attacks the DNA, while RNAi affects RNA expression. CRISPR's method uses a Cas9 complex and can disable the gene or input other sequences, while RNAi just silences the gene using a dicer and RIS complex. RNAi may also silence other genes with a portion of the same RNA sequence, while CRISPR only affects the desired DNA sequence. CRISPR and RNAi can be used in either prokaryotes or eukaryotes, but CRISPR occurs naturally in prokaryotes while RNAi occurs naturally in eukaryotes. CRISPR occurs in the nucleus of eukaryotes while RNAi occurs in the cytoplasm. The scientist then decided to try to limit the expression of the odor gene in his crown imperials using the CRISPR technique. With this method, he could silence the smell gene in the crown imperial, while with RNAi he might have only inhibited it. CRISPR also allows the scientist to be able to alter the gene another way if he wanted to, and limits the chance of targeting a similar sequence and altering a different gene. When the crown imperials grew large and tall, the scientist walked out into his garden and took a big whiff. No foul odor anymore. This method allowed him to silence the gene and keep his garden smelling and looking beautiful.